So hello everyone, I'm Luiz Irber. I'm a computational biologist at the Next Genomics. But today I'll be talking about work that I did during my PhD. Well, so the title of the talk is Petabase Scale Search and Containment Analysis with Fractional Sketch. I did my PhD at the Lab for Data Intensive Biology at UC Davis with all these wonderful people. And this also includes work that we did together with the Kozlicki lab at Penn State and with the Jessica Lumia and the Dawn lab at UC Davis. So the first thing I want to discuss is the flip side of this chart that everyone puts in presentations. The thing I'm interested here is not the drop in cost for sequencing, but what it causes in public genomic databases. So for example, GenBank last year grew 50%, or it grew 50% from 2020 to 2021 to 15 terabase pairs of data. The SRA is reaching 19 petabytes of data. And so the question is, how do we search, how do we use all this data? It's kind of there and it's super hard to use all of these resources available. So I want to approach this problem trying to use lightweight approaches for doing data analysis, especially sketching and streaming approaches, and kind of breaking this process to solve this problem into doing some initial exploration where you subselect the interesting data from these large databases, and then with this smaller subset, you go and do deeper analysis. Especially because not many people have 25 petabytes laying around to download all this data and use. And also, what research questions can we rephrase to be able to use these approaches that are more lightweight for doing data analysis? So as an example, I want to talk about this work that Jessica did for her PhD at the Sumner Lab, where they went to Antarctica and collected samples from a frozen, well, it's frozen on top, but it's a lake called Lake Vanda. And they managed to get the samples and assemble 5 metagenome assembled genome of about five megabase pairs long, each of them. And so, okay, they have this data now. It's Antarctic cyanobacteria. There are no other ones to compare to see like how they are compared to other ones available in Antarctica. So what can she do? Maybe she can take this mag and search through all the public genomic, the public metagenomes in the SRA and try to find other matches for this mag in other places of the world. Turns out that when she did that, she found for these 5 mags that she had, the 2 bottom ones are kind of funny because it's in the same environment, but she found another study done in a lake close by that they didn't know about, and they sequenced the same thing, and it was present on the metagenome that was deposited in the SRA. For another mag, they found it in many different environments, going from polar environments to deserts. And another one is present in harsh environments. And you can see that this one is showing up in a lagoon in France. And this lagoon has a lot of pollution. It has sulfide and hydrocarbon pollution. So this is this mag that they found. It's an organism that lives in very harsh environments. And I'm glad that the GIF actually worked. So for doing that, I developed something called Mastiff, which is a content-based SRA search. So the idea here is that you take your mag, your query, you calculate a sour mesh signature, and then you search against 480,000 metagenomes from the SRA. Performance-wise, for doing 2,600 of mags searching against this database, it takes about 55 minutes using 24 cores and 400 megabytes of memory. And the result for this search is that it returns an SRA accession and a containment score of your query inside a metagenome. And these slides were done 2 weeks ago, so I said sign up for more details. for the workshop on Thursday. You can't sign up anymore, sorry, but there will be a workshop on Thursday with more detail. So I use this measure called containment. So what is containment? I'll use two closely related metrics, which are containment and similarity. I will start with similarity. Similarity is basic, especially Jacquard similarity on this formulation, is measuring how similar two items are to each other. And it works very well when both items that you're trying to compare are about the same size. So what it's doing is that you're taking the intersection of the two items and comparing with the union of the two items, speaking on set terms. A book example for this that I really like this book, so I will use as an example, is the Dispossessed from Ursula K. Le Guin. And the structure of this book is that it's alternating chapters in two different places and time frames. And the first and last chapters are space travel chapters. So what I wanted to check is, do these chapters cluster together when you do a similarity analysis on them? So the first step that we have to do is that we have to break these chapters or these things into sets. And in this case, we are going to break into sets of words. And to do this process, what you do is that you tokenize, you break the chapter into words, into spaces. You can remove stop words like the that don't add any meaning to the content. And you can also do stemming to get the root of the word, for example, to calculate the set. So when I did that, this is a plot showing the clustering of the chapters, and you can see that the odd and the even chapters actually separate. And it's also funny because the first chapter is between both words, so it's traveling, and it also shows up in the middle of the clustering. So this is an example of using a similarity metric to analyze this data. Turns out that when you have items that are not the same size, when one is much smaller than the other, a more interesting question that you can ask is how much of one is present in the other item. And so this is where containment comes in. The containment is a measure, a ratio of the intersection of the two things divided by the size of one of them. So again, for a book example, I'm gonna use the Torah and the Bible, because the Torah are the first 5 books of the Bible. So if you try to compare the similarity between them, it's about 34%. It's not very high. If you try to calculate the containment of the Bible in the Torah, you'll get about the same number because it is all inside. But if you calculate the containment of the Torah in the Bible, then it goes to 91%. So this is a very good thing to do when you're comparing things that are different sizes. Before we go when each one is better than the other, how do we translate this example from books into actual sequencing data? It's not as easy to say what's a word in a sequencing on sequencing data. And even if you say, oh, it's a gene. Genes are very complicated when you're trying to compare many different books or very many different organisms. So we are going to take a step back and break into k-mers. And in this case, it's an example of a trimmer in a sequence. So you do a sliding window and collect all the 3 combinations on this window too. And then the shingling, this very fancy word, is just the process that you do for converting a full data set into these elements of a set so you can do comparisons. How do you choose between containment or similarity? Containment is very good when you have small things against large things. So a meg or a genome in a metagenome, or a config inside a genome. Similarity is very good when you have things of similar sizes. So you're comparing genomes that are from the same kingdom, so like bacteria with bacteria, this works well. We're trying to compare bacteria with eukaryotes, not as well. Chromosomes, usually it's good to compare because they are kind of similarly sized. Or if you're comparing metagenomes against metagenomes. So doing that with the full set, like with all the k-mers of sequencing data, it's kind of expensive. It doesn't save you much space, but there is a technique called min hash, developed around 1997, that was first used to compare web pages in the internet by AltaVista, for those who remember AltaVista. And it provides estimators for the resemblance, what they call resemblance. It's containment is the same. Jacquard similarity is the same thing, and containment. The main benefit of mean hash is that it saves you a lot of space. In genomic context, MeSH from 2016 was the first one to apply this sort of techniques for doing large-scale comparison of genomes. And the approach that they use, or how a mean hash works, is that you take your k-mers, you use a hash function to convert that k-mer into a number, you order all the numbers, and you take, if you're doing a mean hash sketch like MeSH does, you take the bottom 500 ones, for example. If you're doing a frac mean hash sketch, you say, pick all the numbers below a threshold. So this can be vary in size. So for example, for a virus, it will have like 10 hashes. But if you're taking a metagenome, it will have millions of hashes. So it's not fixed size anymore, but it's still a fraction of the original data. Frac mean hash, this approach that we came up in the lab, this alternate way of defining mean hashes, it's implemented in SourMesh, which is a Python library and command line interface with a Rust core for performance. And it provides similarity and containment estimation between data sets. It has documentation, has tutorials. We have a development process with tests and CI and frequent releases for this. And this is a plot now using actual genomic sequence just to show that I actually use the tools from SourMesh to build that plot before with text. So you can even hack a bit and use for other things which are not sequencing data. SourMesh has around 37 code contributors in GitHub. We have many other contributors that are not directly contributing code, but they come up with problems and super nice ideas on how to solve them. And we interact and come up with solutions. So, and there are other methods that implement containment queries like meshstring and cmesh. The main difference between what we do in SourMesh is that in SourMesh you only need the frac mean hash sketches for doing comparison. With meshstring, you still need the original data that you're querying. So if it's a metagenome, you have to have all that metagenome lying around. And for cmesh, it's a Bloom filter, which is a compressed representation, but it's another data structure involved. If you compare all of them to the ground truth, so if you take an exact approach and try to see how the estimators work, they all work great. So the meshstring and cmesh are great tools also. But the main benefit is that because the frac mean hash grows in size, it can adapt better to like how much complexity you capture. And it's kind of reflected if you go and calculate frac mean hash sketches for GenBank domains. You can see that viral sketches on a scale of 1,000 are pretty small, and it kind of goes growing as the complexity of the genomes increase. Yeah, so the benefits of frac mean hash is that it allows efficient containment queries. Without that access to the original data, you can do all these comparisons using only the sketches. You don't need any auxiliary data structure, so you can do manipulations, again, using only the frac mean hash sketches. And you can do subtractions and abundance filtering in these sketches too. And there is this scale parameter, which is kind of a tunable trade-off between the precision that you get and the resource consumption. So if you want more precision, you can use more memory and more space, and you get better results. And it also works like if you're working with viruses, you need to go to a higher scale value to get more hashes back. But if you're dealing with metagenomes, you can get to higher scale values like 10,000 or 100,000 to save space and do the comparisons quicker. So we have frac mean hash sketches. Wouldn't it be nice if you don't need to download all the data and calculate sketches, if you can just use the signatures, the SourMesh sketches? So I came up with a system back in 2018 that would take requests for calculating sketches or these SourMesh signatures from public databases. And I organized kind of as a test queue that received these requests, and then workers go and pick the jobs and download the data, calculate the signatures, and put on an S3 bucket. It's been chugging along since 2018. So nowadays, again, these slides are from 2 weeks ago. We surpassed 13 terabytes of SourMesh signatures. It's about 4.7 million data sets that are available, the signatures for use. And the other thing here, I kind of broke into single genomes and metagenomes because of the way I was searching the SRA for calculating the signatures. And it actually surpassed a petabyte of original data. So the petabase scale on the title of the talk, it's actually accurate because I did index all the more than a petabyte of data from the SRA. And it's not only the SRA, the Word also has signatures for GenBank, RefSeq, and IMG. So back in 2020, I was writing my dissertation and I had all this pile of data and I didn't really have a use case for it. So in about a week, I concocted something that I call mag search, which is this idea that you take a mag that you sequence, you search through the SRA to find where things are. The first time I tried this, it was with the 2600 mag queries from the Tulip paper from 2017. And I found about 23,000 matches above 50% containment from 6,300 unique SRA runs, discounting Tara and other data sets that were used for assembling the mags because I didn't want to cheat. I wanted to find new things. And this took about 11 hours running on 32 cores and 12 gigs of memory. And the idea was kind of, I didn't have any good sour mesh indexes that would work with all the 700 gigabytes of metagenome data. So I kind of just loaded them all in parallel and compared with the queries. So this is high latency because I had to load all these metagenomes if they had matches or not. So the time for doing one query is pretty much the time for doing 2,600 queries. So Massive is kind of the next step on this lineage. And it was something that about 2 months ago I started implementing because I had this idea in the back of my mind that I could do this using a disk index and not a memory index, and then I could actually build this thing. And it turns out that it worked. So again, the GIF running on real time. And before, it was taking 11 hours for running through these 2,600 mag queries. Now it takes 55 minutes using 24 cores and 450 megabytes of memory. And takes about 10 seconds per query, and that's the latency. It's if you start the search, you get results in 10 seconds. You don't need to wait 11 hours to get results back. There is a client available that can do the conversion from raw data to signature for you and submit to the server that's running with the index. And then this command on the bottom, it's actually downloading RefSeq signature from Word, feeding into Massive, and then getting results. So if you're interested in something that's on RefSeq or GenBank, you don't even need to download the genome to be able to run. I kind of have 1 minute, so I'll go quickly through this. For validating these results, I took one of the mags from the Tulip paper from the North Pacific and trying to find in other places. When I was preparing the slides, I did a search for this mag, and I actually found that there is a paper from last year where they did sequencing in other places around where I found a match back in 2020. And it's the same species. Like this thing is actually present in the South Pacific, which I think it's super cool. You can also use this for outbreaks. This is a paper that tried to do the analysis of an outbreak, and they use this kind of search through all the metagenomes. And they actually found a match in the US while this outbreak was happening in Europe. They weren't expecting to find in other places, and was just on the SRA, like on a metagenome dataset. I'm on time, but there are some other tools in the lab also using sour mesh. You can do taxonomic profiling. It compares very well with other tools. You can do this in the browser. Actually, I will implement this front-end interface for Massive also, but it will calculate the signature for you in the browser and send to a server to get back results. You can do contamination detection in genomes using this approach for decomposing queries into their components. Genome Grist is a workflow that automates this process of downloading data of the SRA once you have matches and doing a bunch of nice plots and summary information. And finally, there is also this fast clustering of hundreds of microbiome data sets that the next version is going to use. the sour mesh signatures, not sour mesh, but showing that track-me hash is also available and useful in other places which are not only sour mesh. And that's kind of it.